All right, let's get right into it. We're talking about something that sounds like it's straight out of science fiction, but it's a very real quest in medical science. It's the search for an implant that can fix you from the inside and then, when it's all done, just disappear.
Seriously, just think about that for a second. You have a bad break, maybe in your leg. Surgeons go in, they put in a metal plate to hold the bone together while it heals. But instead of that plate being there forever, it just slowly and safely dissolves away as your own bone gets strong again. By the time you're healed, it's gone.
And the reason this is such a big deal is simple. Right now, with titanium or steel implants, they're in you for life, unless you get another surgery just to take them out. This new idea completely gets rid of that second procedure. That means less risk, less pain, and honestly, a whole lot less cost for the healthcare system. This isn't just a neat trick, it's a total game changer for how we approach healing.
Now, to pull this off, scientists have had their eye on one particular material. It's almost perfect, and I mean almost. Let's meet our hero material, and its one single fatal flaw.
The material is magnesium. It's got the strength we need, the body tolerates it well, and here's the magic trick. As it biodegrades, it actually releases ions that encourage your body to build new bone. It's like a construction scaffold that not only holds the building up, but also shouts encouragement at the construction workers.
But, of course, there's a but. And with magnesium, it is a really big one.
The problem is, on its own, magnesium just dissolves way too fast in the body. The implant could literally fall apart before your bone has a chance to heal. And what's worse, the chemical reaction of it dissolving creates hydrogen gas. We're talking about tiny bubbles forming right where new bone is trying to grow, getting in the way and preventing a good solid heal.
So the challenge becomes pretty clear, right? You've got to slow that whole process down. And the solution scientists came up with is actually pretty elegant. They decided, why don't we just give the magnesium a little suit of armor, a microscopic shield? So they came up with two different kinds of shields. The first one, called a hydrothermal or HT coating, creates this really cool, spiky, needle-like surface. The second one, a sol gel or SG coating, is totally smooth. Now both are made from a material the body is totally fine with, but that smooth one, the sol gel, had a secret ingredient, a tiny bit of copper, which they hoped would help fight bacteria and maybe even boost bone growth.
Okay, so we've got our shielded implants. The next logical step is to see how they hold up in the lab, in a fluid that's designed to mimic the conditions inside the body. And let me tell you, the first round of results, they were amazing.
Whoa, okay, just look at this chart. This shows how fast the different implants corroded. The uncoated magnesium is chugging along at a rate of 181. The smooth sol gel coating helps, it brings it down to 135. But then look at the hydrothermal coating. It drops the corrosion rate to 0.03. I mean, that's practically zero. In the lab, this looked like an absolute, undeniable home run. The shield was working perfectly.
And the good news just kept coming. This chart here shows how many living cells survived when they were put right next to the material. With the plain, uncoated magnesium, it was pretty toxic at first. Only about 20% of the cells made it. But with both of the coated implants, cell survival was way above the 75% safety line. So the shields didn't just slow corrosion, they made the implant completely safe for our cells.
And the cherry on top, a crucial safety test, the Ames test, came back completely clean. This means none of the materials were causing any dangerous genetic mutations. So, you look at all this lab data, and what do you think? It seems like they've cracked it, right?
But science is rarely that straightforward. Because what works perfectly in a sterile, predictable lab dish, well, that doesn't always translate to the messy, complicated, and super dynamic environment of a living body. The story was about to take a sharp, unexpected turn.
So, let's just recap. After seeing all that incredible lab data, the conclusion seems obvious. These coatings, especially that spiky hydrothermal one, are the perfect answer. Problem solved. End of story, right?
Yeah, not so much. When the scientists put these implants into actual lab animals, the first major surprise was how well the new bone attached itself directly to the implant. And check this out. The plain, uncoated, problematic implant, it had the best bone contact. Both of the supposedly superior coated implants had way less direct connection to the new bone.
So what was going on here? Well, when they looked closer, they saw the shield was almost working too well. It was creating this tiny little barrier, a gap between the metal and the new bone. And in that gap was this layer of kind of sort of bone, but not the mature, strong stuff you want. The shield was preventing that perfect, strong integration. But that wasn't even the biggest shocker. Let's look at how much the implants had actually dissolved after 12 weeks inside the body. And this is where things get really wild. Look at the Sol-Gel implant, the smooth one with the copper. Almost 40% of it was just gone. Way, way more than the uncoated implant it was supposed to be protecting. I mean, just let that sink in for a moment. The coating that was specifically designed to protect the implant from dissolving actually made it dissolve faster inside a living animal. It did the exact complete opposite of what it was supposed to do, and the total opposite of what every single lab test had predicted.
So what on earth happened? Well, it was a perfect storm of chemistry meeting biology. First, remember that tiny bit of copper they added? It turns out it acted like a bunch of microscopic batteries on the surface, creating little hotspots that sped up corrosion. But the real kicker was the body itself. In the lab, as the implant corrodes, it changes the pH of the fluid around it, and that naturally puts the brakes on the reaction. But inside the body, you have constant blood flow. It just swoops in and washes all those chemical changes away instantly, basically removing the brakes and letting that copper-fueled corrosion run completely wild.
So look, this isn't a story about failure, not at all. It's an incredible story about just how complex our bodies are, and it's a perfect lesson in why you can't stop at the lab. You have to see how things work in the real world. So let's break down what this really teaches us. This little scorecard just sums up the whole story perfectly. In the lab, the coatings were the champs, no question. But inside a living animal, the uncoated implant had the best bone contact, and the promising Sol-Gel implant just fell apart the fastest. It just goes to show you that how you measure success really, really matters.
And if there's one thing to take away from all this, it's this right here. A simple lab test is a vital first step, but it just can't replicate the absolute beautiful chaos of a living system. Your body isn't just a beaker of salt water. It reacts, it adapts, it pushes back, it changes the rules of the game.
And so this whole journey leaves us with this one profound question. The challenge isn't just making a material that dissolves. The real challenge is making a smart material, one that knows how to be strong and supportive for exactly as long as the body needs it, and then knows exactly when it's time to gracefully and quietly get out of the way. That, right there, is the true quest for the vanishing implant.